I guess the good news is the first speaker, there's no chance of being redundant with anyone else. The bad news is I don't have a real good feeling for where people's heads are at the room, where your backgrounds are, and so on. I was told to give sort of a primer in population genetics for the first lecture, maybe lecture and a half, before we merge on into quantitative genetics. I'll have a little bit of both in my talks, but this is sort of the roadmap where I'll be going. First, I want to just talk about the noise in the evolutionary process, this concept of random genetic drift and effective population size. I'll spend some time talking about mutation. It's a source of variation on which natural selection operates. Then we'll start to put two things together. What happens when mutation and drift are interacting? Three things together. What happens when mutation, selection, and drift are interacting? Finally, four things together: recombination, drift, mutation, and selection. I have a feeling that my lecture today will only get through the first part. So we'll see how this goes, but feel free to ask questions as I go along. As I said, I don't really fully understand everybody's background. So just to get started, I wanted to point out that evolutionary biology is a really broad field, and lots of people are doing lots of different things and operating from different ways of thinking and so on. For a lot of people in evolutionary biology and in breeding, this is sort of what you're interested in. You're interested in directional response to selection, making things bigger, smaller, faster, slower, whatever happens to be your target destination. And this works really well without even really knowing any genetics. So I guess in 1957, people were eating really tiny little chicken sandwiches, and now they're eating these gigantic things that you can buy at these fast food places in the United States, at least. So dramatic improvement in the amount of biomass in chickens. And the plot on the right is increases in grain yield in hybrid corn over the past 80 years or so. And the three lines there are just for growth. This is based on stored seeds, so you could raise everybody in a common environment. This is just showing there's been essentially a linear response to selection over time. All this is done without knowing anything about the molecular genetic basis of these traits. And then, sorry, it'll take me a second to get used to the clicker here. And then there's another whole field of biology that's interested in sort of the equilibrium distribution of mean phenotypes across very, very vast sets of taxa, the whole tree of life. And these are just three examples of various scaling relationships that we've come to learn. The one on the left is a scaling between metabolic rate and dry weight of organisms. There's several orders of magnitude we're looking at here, and it really starts to bend and slow down as you get into multicellular species. The middle one's a plot of the average growth rate, maximum growth rate of organisms, function of size, different rules in bacteria and eukaryotes. And then the one on the right is a measure of swimming efficiency for energetic investment across the tree of life. So all these are general scaling relationships that people have observed, but this is done completely in the absence of thinking about genetics. Usually they're engineering sorts of arguments that are made to explain these things, and I'm not sure that those are always the appropriate arguments. So the way I like to think about evolution, it's sort of a triad of triads. So down at the bottom is what most evolutionary biologists are thinking about, things going on outside of cells. You know, finding mates, finding resources, food, and avoiding bad sorts of things. Cell biologists have taught us a lot about what goes on inside cells, and this turns out to be quite important and quite useful, trying to understand the basis of environmental variation between cells. So the variation among individuals in the absence of any genetic variation. We're not allowed to break the laws of biophysics, apparently. And historical contingencies turns out to play a really important role of what natural selection can and cannot do. It's descent with modification. But ultimately, what selection can't and can't do is dictated by what I call the population genetic environment, which has three dimensions. First is random genetic drift. This is the noise in the evolutionary process that I'll attempt to explain. Mutation, I'll talk about later, as I said, source of variation upon which selection operates. And recombination reassorts variation within and among chromosomes. In some cases, it may be good for adaptation, other cases, bad. we're interested in these kinds of things when you're thinking about evolution across the whole tree of life, because all three dimensions of the population genetic environment vary by orders of magnitude across the tree of life. So on the left, something I'll get into soon, but just to give you a prelude, this is the negative scaling relationship between what we call effective population size and the size of an organ in the adult weight. And it scales with an exponent of about minus 0.2. So if you increase the size of an organism by 5 orders of magnitude, or 10 orders of magnitude, you'll in general decrease the effective population size by 2 orders of magnitude. The middle one I'll talk about more later. This is a scaling of mutation rates in organisms as a function of the effective population size. Scaling relationship there is -1. You can see there's variation by about 4 to 5 orders of magnitude in the mutation rate amongst different organisms. And then finally, the plot on the right is a relationship between the amount of recombination per nucleotide site in genomes and the size of a genome. And this has got a slope of about -1 on average. This is simply due to an almost indelible rule in eukaryotic genetics, is that during meiosis, there's typically just 1 or 2 crossovers per chromosome arm per generation. So you double the size of the genome, you're reducing the amount of recombination per physical distance by 50%. Yep.
So you're showing this algebraic relationship between these two parameters, between the y-axis and x-axis.
Mm-hmm.
Is there any deeper reason why we should expect all these algebraic laws and just a fit? Because it happens to be
Can we explain these from first principles? Is that what you're asking?
No, I mean, why power laws?
Why power laws?
Did we have some other
Well, I view power laws as a first approximation, but these look pretty linear on a log-log plot, right?
First one?
Pardon?
First one. The slope -0.2.
Yeah.
Could we have some other functions which are better fits? Have they been tried?
Those are data from me. I haven't really tried. I guess I was pretty satisfied with the linear fit there on the log-log plot. I suppose you could add make it a polynomial, but it's hard for me to see that would change things very much. But it's worth entertaining. I don't mean these to be indelible laws. These are first approximations of the way the data look. I'm just trying to present the fact that these major aspects of the population genetic environment all vary dramatically depending on what kind of organism you are. And I'll go into all 3 of these plots in more detail. I'm just trying to get the big picture out right now.
I think the metabolic rate is a function of the size of the organism.
That's not linear. Within groups, it's roughly linear, but then as you move to larger and larger organisms, it starts to curve. That'd
I
be.
think there's some theory for it being.
There is some theory that we don't all agree on, but there's a lot of thought given to some of these things, especially for the non-population genetic parameters. Metabolic rate has probably attracted more attention than almost any other trait in biology.
But is the one on the right, are there bacteria on there?
No, this is eukaryotic. That's all eukaryotic. It's a crossover recombination rate based on.
And where's the rate on bacterial?
I'll probably talk about that a little bit later, but bacteria have scaled to the mutation rate. They're more or less the same as you find for eukaryotic recombination rates. Bacteria have quite a bit of recombination. It's just different kinds of recombination. Okay? Any other questions at this point? Because I'm just getting started, so. Yeah, I'll talk about this more in a few minutes, but even bacteria almost never exceed an effective population size of 1 billion. That's a tiny pile of bacteria. And I'll try and explain why that is. It's not just a numbers game. There's a lot of other stuff going on in terms of chromosome structure. And then this is sort of a guiding principle that'll underlie a lot of what I have to talk about today. There's this concept of what we call effective neutrality. If the magnitude of selection operating on nucleotide variant is smaller than the power of random genetic drift, then the noise in the evolutionary process just wins. And Tomoka Oda was one of the first people to point this out. And so this means that even mutations that are truly non-neutral will act as though they're neutral in certain kinds of situations if the effective population size is sufficiently small. And so we're going to be thinking about genes and mutant alleles moving through populations like that red ball up there, that's a new mutation. That happened to eventually go to fixation by some mechanism, either pure drift or selection. And so the key here is, let's just think about haploid organisms here. And there's two alleles, the famous big A and little a allele. And we can benchmark one as having a fitness of 1 and the other 1 plus S, where S is a selective advantage or disadvantage if it's negative, usually a fairly small number. And that's going to become important because if S is small and NE is sufficiently small, then selection can't see things adequately to move things forward. So this might be viewed as a real evolutionary law. If the selective advantage or disadvantage of mutation, let's say we're focused on the absolute value, is less than 4 over the times the effective population size, selection is rendered ineffective by the noise in the process. So a lot of what goes on in evolution is not just about the absolute value of different population genetic parameters, but it's dictated by their ratios. And that'll come up again. So just to demystify this process of random genetic drift, and I guess I should apologize at the outset. I suppose there's physicists in the room, and I know that physicists use the word drift in very different ways than we use it in biology. I believe in many formulations in physics, drift is the directional term, and here we're using the word drift as purely a stochastic force of evolution. So what's going on here is, in this cartoon view here, we have a very small population of just 10 individuals up here. Let me see if I can get the pointer working. And then those 10 individuals produce a gamete storm, but the environment can only hold 10, so we sample 10 random balls from here. And you see the allele frequencies are 50-50 here, and after one generation, it's bounced to 0.6, and then we repeat this and repeat this, and these numbers are bouncing around. And eventually, over time, we'll lose one of these types or the other, unless they're maintained by some kind of balancing selection. But right now, I'm looking at the pure case of random genetic drift. And what's important to realize is this process occurs in all populations of all sizes. It's just the magnitude of the fluctuations is inversely related to population size. So here we're looking at, again, starting at allele frequency of 0.5. This is just the trajectory of the allele frequency in one of the populations, the population size of 25. And eventually this goes to 1. We call that fixation of the mutant allele. And if it's neutral, it could have just as easily drifted down in the opposite direction and been lost. And now we go to a population size 10 times larger, and you see there's still fluctuations. Eventually go to 1 or 0, but the fluctuations have been muted by a factor of about 10, and then we increase it still further, and we're just increasing the time scale at which things unfold by the factor that the population size is increasing. This is the ideal case in which the only thing matters is the numbers of individuals. And the other thing to emphasize, I've tried to already make the point, is that these are just one realization of a random process. So if you started multiple replicates and everything's neutral, half of them eventually go to fixation, half of them eventually be lost. And it's always nice to have real data to demonstrate things. And so this is these are results from a famous experiment by one of Sewell Wright's students named Burry. I think he was so exhausted after these 2 experiments, I don't think he ever published another paper. But it was quite a piece of work, and it's a beautiful textbook example of random genetic drift. He started with populations of flies that he had 16 flies, 8 males and 8 females, and they were all heterozygous initially for a color marker allele that he could just, this is in the pre-molecular days, so he could genotype the individuals by looking at the eye color. And after so they all started here, and after he had 256 replicate populations, by the way. And so it's actually in 2 experiments. I don't think he could have physically done 256 all by himself at once. But you can see after 1 generation, the allele frequencies have drifted away from the initial value. And after 5 generations, they drifted still further. After 10 generations, there's almost equal frequencies, equal probabilities of the replicates having a particular frequency. But now you can see that this bell-shaped distribution is becoming more of a U-shaped distribution. And that's because eventually the populations are all either losing one allele or the other. They're becoming fixed for the mutant allele in a random fashion. This fits the theory in quite nicely. It's quite consistent with what was expected according to theory, but there's one neat twist to this experiment. It'll start to maybe help you appreciate all the nuances that affect effective population sizes. So we have this simple formula here that if the population is behaving the ideal way I just outlined in the previous plots, just random sampling random balls from an urn, if we have a diploid individuals and there's n individuals, there's 2 n copies of the alleles at the locus, and this is the heterozygosity. It's the fraction of individuals in the population that have 2 alleles. And if you let things go long enough, this is the decay rate of the initial heterozygosity over time, and we can approximate it as that. So it'd be this exponential decay at rate 1/2n. But then when you plot the data, you see that sort of looks like that, but it's not exactly like that. So this is the theoretical expectation if you plug in 16 for n. There are 8 males and 8 females always drawn, but the heterozygosity on average is decaying more rapidly than expected according to theory. And to make the theory fit, then you have to use an n of 9. So the absolute population size was 16 generation after generation, but it was effectively behaving as though there were only 9 individuals. There are lots of reasons this could be. Probably the main one would be variance in family size. Some individuals are more prolific than others, and that just sways the balance to a smaller effective population
Eggs
size.
are not selected for pair of parents. Eggs are selected randomly.
I think he was selecting, if I recall, Bruce, do you remember? I don't think he was selecting at the egg stage, but maybe.
No, but I mean, the eggs are chosen randomly.
Yeah, everything's chosen randomly. There's no selection here. That's
No,
important.
You could try to choose 1 egg for parent.
They're chosen randomly from a mixed
Oh, okay.
batch of propagules. Yeah.
And you grab 8 males, 8 females, make the next generation. So the actual size is larger than 8 males, 8 females.
Yeah, this could get even trickier. If everything was random, if there were, he picked Poisson distributed random family sizes, then it presumably would obey the theory. If you picked exactly 2 from each, that would actually increase the effective population size. This is hard to get your head around, but it would double the effective population size relative to the absolute size. And this is used often in conservation genetic breeding programs as a way to maximize effective size. Anyhow, I'm just trying to get at some of the demographic issues that are important here. Now, that's a tiny population. You might think, well, natural populations, unless they're endangered species, are a lot larger than that. And in fact, they typically are. So this is a plot from several years ago by Finlay. This is the scaling of the number of individuals per meter squared as a function of organism size. And you can see that there's this bigger things typically have smaller population sizes. That won't surprise you. But this is per meter squared, so you'd have to multiply that by geographic range to get some idea of the absolute head count. That's actually been done in some cases. Lately, Ron Milo's lab has been trying to summarize enormous amounts of data to get an idea of where biomass is distributed on the planet and so on. And these numbers here are the number of individuals, insects in boreal forest, temperate forest, and so on. Those are really big numbers. And even if we divide by, say, 1 million species to get the average number per species, we're left with still a really big number. These are the number of individuals of some mammals that constitute most of the mammalian biomass on the planet, other than our own and cows, I guess. But you can see that those numbers too are really big. So should we worry about effective population size if the power drift is 1/2n? Well, it turns out that let me back up a second. I need to get through the definition of effective population size. And the thought process behind developing formulations based on demographic features of populations is related to this concept of identity by descent versus identity in state. So imagine we have 2 parents, and this parent has 2 different alleles, the white one and the black one, and then it's going to pass some gametes on to progeny. These 2 gametes are copies of the same parental allele. So those are, by definition, identical by descent. This one over here, it's a black ball, but maybe it arose by an independent mutation. So it's identical in state to these over here, but not necessarily identical by descent. And then this one's clearly a different allele. And so the way to think about this is, the way to think about the definition of the effective population size is its inverse is the expectant fraction of zygotes in a population. By a zygote, we mean two gametes coming together to make a diploid individual. It's the expected fraction of zygotes derived from the same parent. And if the two alleles in a zygote are derived from the same parent, there's a 1/2 chance that they came from the same allele in the same parent. And then you can write down recursion equations like this that explain the increase in the degree of identity by descent over time. And so there's a 1/2, that's the probability of drawing alleles from the same parent, and half of the time they'll be identical by descent. If they're not identical by descent immediately, they might have been identical by descent from some preceding generation and so on. I'm just trying to give you a general flavor for what people are trying to get their heads around when they're thinking about the concept of effective population size. And what people do is they think about various mating systems, sex ratio, variance in family sizes, all kinds of things, and try to think if that's the state in the population, we're drawing random gametes for the next generation. Given that kind of demographic set of features, what's the probability they're identical by descent? And just as one example I've given here for a monoecious or a hermaphroditic population. So each individual produces male and female gametes like many plants do. And you can work this out analytically. And here the effective population size depends on 3 different things. So it's nice to see it's positively related to the actual number of individuals in the population. That might be intuitive. But it's also related to the variance in the number of gametes, the variance in family size among individuals, and the mean number of progeny produced per individual. And so this can even further simplify. If we assume the population is stable, then everybody has 2 kids on average, and we plug the 2 in here, and that's our formula. And this nicely shows that the effective population size declines with the variance in family size among individuals. There's lots and lots of formulas like this for different kinds of breeding systems. And I won't go into those, but I just want to sort of summarize what influences demographically effective population size. And almost everything you think about demography reduces any relative to the absolute head count. So as I already said, variation in gamete production, that could be due to selection, will cause a reduction in the effective population. Population subdivision and variation in productivity among subpopulations will also influence effective population size. If there's temporal variation in population sizes, which there always will be, the long-term effective sizes comes to resemble more closely the bottleneck size as opposed to the flush size. And then deviation of sex ratio from 1 to 1 will also influence things because half the genes in the next generation come from one sex and half come from the other. So the rarer sex will really determine the overall effective population size. So all these things are demographic aspects that influence the effective size. It turns out that this is probably not, especially in species with large number of individuals, these are probably not the major influencers of effective population size. The very structure of our genome really influences what goes on in terms of the maintenance of variation in population. So imagine here, we're looking at a population. These are chromosome segments, and all the tick marks are polymorphic sites, and presence, absence, polymorphisms in the population. And that chromosome there happens to have picked up a beneficial mutation, and it's so good that it has swept through the population. And some generations later, all these other chromosome types are left or removed from the population. They're displaced by the one successful chromosome. So that's good, but 2 other things have happened here. So that little tick mark to the left there, what I've illustrated now with the red ball, that turns out to be another beneficial mutation. And so it's not as good as the blue ball apparently, and it's eliminated from the population because this chromosome segment wins. And then finally, there's one other tick mark there that I meant to illustrate, the green one there. That's a deleterious mutation. It's not as bad as this one's good, but the net selective advantage of this chromosomal segment is good enough to allow it to drive through the population. So what's happened here, because of the lack of recombination in this region, we have a situation where we fixed only one of the two beneficial mutations of this region, and we've lost a we've also fixed in the process a deleterious mutation. If we had free recombination, selection could operate on each of these sites independently of each other, and we would have fixed both the beneficial mutations and lost the deleterious mutation. So that's one of the many consequences of recombination or lack thereof in influencing the efficiency of natural selection. You can see these sorts of things in laboratory populations in which large numbers of individuals are marked. And this just happens to be a set of replicate studies done in yeast, where yeast are just figuring out how to grow more rapidly in a test tube. And you can follow the individual clones over time. And you can see in asexual populations, you'll see these big bundles. Each one of these is a polymorphism going to high frequency. And you can see in asexual populations, there's a whole bundle of fixations all at once. This is because these are clonal populations. And this clone has apparently picked up a highly beneficial mutation, but it dragged everything along to fixation with it.
I mean, it probably picked up several smaller ones.
Possibly, yeah, yeah. But it's a whole linkage group is the main thing. So you get a whole constellation of things. Many neutral ones are probably fixing, some deleterious.
Neutral rates.
Maybe. To the right, you don't see these bundles is the main point. And there's recombination that was enforced regularly in these populations. So that's all I'm trying to make the point. You can see these principles I'm trying to point out with real data in laboratory populations these days, now that we're able to do high throughput sequencing in populations. Now, possibly the
Can I just ask, because I've never understood why effective population size keeps getting called effective population size. If I had data like that, how would I get the effective population size from the data?
Well, you would have like 6 different effective populations.
Yeah, I was hoping to hold this to last. Well, just as a prelude, there's multiple ways to define effective population sizes too. What I'm focusing on now is what might be called a variance effective population size. It's the NE that's associated with the maintenance of variation in populations. I'll try to explain this in a second. I don't see why it couldn't be applied here, except these populations aren't in equilibrium, and the methods I'm going to be introducing are assuming populations in equilibrium. So a second issue is this, and maybe even more important one than the previous slide, and this is that the vast majority of mutations that we'll learn later are deleterious. And so what you can imagine now is a situation where you have all these chromosomal segments, and some chromosomes have accumulated mildly deleterious mutations over time, and they're deleterious enough so they'll eventually be purged from the population, leaving only these other cleansed chromosome segments behind. And so that also operates as a form of internal selection, but reduces the effectiveness of transmission of genetic information to the next generation. And you can also look at this. One prediction of this theory is that this will be less of a problem in regions of high recombination. And these are some results from Michael Nachman's paper quite a few years ago in humans. And this has been shown in other things like flies, is that genes in regions of low recombination tend to maintain lower levels of variation. All these chromosomal regions are subject to the same mutation rates, the same rate of mutation coming in. But if you're in a low recombination, then you're more likely to be purged due to these linked deleterious mutations. This is called background selection. And probably we'll hear a lot more about many of these things in a more rigorous manner throughout this course, but people are interested in these kinds of issues about purging of genetic variation by various forms of selection. So the first thing I was talking about is effectively a hard sweep. You get this complete elimination near the region of the selected locus. You can have a soft sweep where the site of interest under selection may go to fixation, but there's also recombination, so you don't lose all the other variation. This is something that we'll probably learn more about later with polygenic selection, where the selection is distributed over a very, very large number of loci. And then finally, this is the background selection hypothesis. It sort of predicts assuming constant rates of recombination, just an overall lowering of the amount of variation maintained in populations. So I've been referring to standing levels of variation, and so we want to make the link here. We actually use standing levels of molecular polymorphisms in populations as one way to estimate the effective population size. And a gift from the gods to us is there's, I think, 7 amino acids that, due to the redundancy of the genetic code, in the 4th position can accept an A, C, G, or a T. That's all the letters we have in our DNA vocabulary. And you still get a threonine in this particular case. So those are called silent sites or synonymous sites. And then out here, that would be a replacement site. If you change the A, you no longer have threonine. And we're going to focus on the silent sites right now and try to think about how that can be, information there can be used to estimate the effective population size. And so the way to think about this is that there's some standing level of variation. We'll call it nucleotide heterozygosity. It's the fraction of random pairs of alleles brought together that are different. And there's going to be some effective population size that we're interested in estimating here. And that's the rate of loss of heterozygosity. So that's the level of maintenance of heterozygosity after 1 generation. And then we have drift then removing heterozygosity at that rate per generation, but mutations bringing in new variation each generation. There's 2 alleles being compared. So 2U is the rate at which new variations introduced. And then you can write down a recursion equation for the heterozygosity in the population. And you can solve this by setting the heterozygosity at time t plus 1 to that at time t. And that's the equilibrium level of heterozygosity in the population. The point at which the input of new variation by mutation is balanced by the output by random genetic drift. And when you do that, this is the formula you get here. And this thing called theta here is the quantity for NE. For NEU. So one way to think, again, we're looking at ratios of different population genetic effects. The power of drift is 1 over 2 NE for a diploid. And so we flip that over, we get a 2 NEU. And I won't go into where the additional 2 comes from. But the point here is that the standing level of variation reflects the balance between mutation and random genetic drift. The formula has got this other term down here, 4 times theta, making it a little messier, but usually we ignore this term here. And the reason is because theta is typically much, much smaller than 1. So as a first approximation, the standing level of heterozygosity is equal to this quantity theta. So that means that if you can measure standing variation at silent sites in populations, you can equate it to this quantity here. So that's the good news. The bad news is this is the ratio of 4 times the effective population size times the mutation rate. So if you want to get an estimate of NE, you need to know what the mutation rate is, and then you can factor it out. And in fact, as we'll see later, my lab is...
Can you just go back?
Sure.
Because this is where I get confused about the focus. So why not just define NE as theta over 4 U?
You could.
That's
Well,
directly
theta's the, if you want to equate that to H, that's...
Theta is an observed quantity and measurable quantity, and mu is an observable quantity. NE
right.
NE is an inferred quantity.
It's an inferred quantity, yeah.
So we can, for most purposes, think of it as just being theta over 4 mu.
Yeah, that's what I'm going to be doing, I think. Yeah.
I mean, another way to think about it, the way to think about all these population checkings is time. Why do you want the rates? Well, because if drift occurs, if change in allele frequencies faster than selection changes, drift will win. Drift time scales is 2n, selection time scales is 1 over x. And so a lot of these different ratios, like recombination, mutation, arise because you're basically competing rates. How long does it take something to happen? When you have a sweep, what happens is the allele frequencies are moved much quicker by selection than they are by drift. You see a different pattern. And we'll see that you think about that underlying feed, a a lot of what's like complicated population stuff. To a first approximation, you can make it more intuitive by just simply thinking about what's the speed up or slow down the rate of this process or that process.
But if I think of any as just being a time and generation in which something will randomly fix, which could be coming from selection, demographics, dynamics, all kinds of things.
It can be related to time, as I'll get back to in a few minutes. I guess the students might not immediately see why this is. Do we have any questions from the students in the audience? Yeah.
So I think one thing which I worry about is that how do we know if we have a mutation, if balance has reached? We often use this formulas.
Yeah, that's a very good question. So the theory that I'm going to be, well, I've introduced the theory, and I'm going to present some data, but the estimates of NE do assume that the population's in drift mutation equilibrium. We're assuming the sites are not under selection, but also that there hasn't been a massive demographic change recently, just changing things. So these are always things to worry about. I mean, this is a real constraint we have in evolutionary genetics. This is the only way we can measure effective population sizes. This is the only one of many kinds of effective population sizes as well. Nick?
This is a null model.
What?
First approximation, I should just think of it as a null model.
Yeah, I'm trying to present the simplest possible model at this point.
Not like out there, Mike.
I don't know if I would call it a null model. I mean, it's a null model of maintenance of variation under various assumptions, one being equilibrium, okay? And we assume the mutation rates also stayed constant as well.
So I think a simple way to look at it, without having to see equilibrium or anything like that, is just to say that what ANE is doing is the expected time when two randomly chosen genes share the common ancestor. Because then, if you're assuming neutral mutation, then we have to have transforming genes. That's it. And the point is that the time when they share the common ancestor, they do all kinds of stuff. And that's complicated. So maybe we should think of already as a time scale of common ancestor.
Yeah, excellent point. And I think Dan was trying to make the same point. And it might not be apparent yet why this is true, but hopefully by the time I'm done, if I'm ever done, there is a connection between what we call the effective population size and the depth of the genealogy in a population. That's essentially what you're saying.
I think
Is
the
that
more
right?
important thing is biology is not static, these won't change. So just to mess things up even more, what human geneticists and Calgary have done is you look at something we'll talk about called Lynch disequilibrium, and you can actually estimate what the effective population size was at different times in the past by looking at different regions of LD. So NE changes over time. If you go through a bottleneck, NE recovers certain features but not other features. But I think the bottom line here is use what I call modestly Walsh's principle. Don't think too hard and you'll get the first approximation. Then we can worry about all these really interesting next levels of subtleties that come on top of that.
Could you write that down? I need to use that quote later. Okay. Anyhow, don't think too hard, but do think. Now, that's the theory. And as I said, if you measure standing variation and you happen to know the mutation rate, you can back calculate, infer what the effective population size. And I've already shown you this graph before, but that's where these numbers come from. These are all from everything from bacteria to vascular plants to vertebrates. And these are all organisms we have measures of a standing level of variation at silent sites. And we happen to know the mutation rates from a lot of hard work that a lot of people have done over the last few years. And a couple points I want to make. This is connecting us back to the noise of the evolutionary process and the efficiency of selection. So if you happen to live way down here, which is where us crunchy things live, us vertebrates, we're way down here, your effective population size, let's say, is 10 to the 4th. And that means the power of drift is 10 to the -4. That means that any mutation with a selective advantage or disadvantage less than 10 to the -4 can't be easily perceived by natural selection. So those are mutations that can't be utilized by long-term selection. And then if you happen to live up here, or if you happen to be a bacterium, interestingly, based on standing variation inferences, that's you know, if we want to push it to 10 to the 10th, that's as big as effective population sizes get, even for bacterial species. But what this means is that if you have an NE of 10 to the 9th, you can utilize beneficial mutations with effects as small as 10 to the minus 9, certainly 10 to the minus 8. And you can purge deleterious mutations with effects that small with the other sign. And so what this means for evolution, there's something to think about here. I guess often most quantitative geneticists are thinking about big things down here, land plants and animals. But if we think about things across the whole tree of life, you expect evolution to be more granular in nature when you're living down here than when you're living up there. Bacteria can take advantage of a broader range of evolutionary sources. And we don't really have, the problem here is we don't have, yeah, sure. Mm-hmm. Well, if a,
The problem is, in that situation, all other quantities you can measure, except for hydrogen, will be different than they will in. One has to go and push this and say, okay, which other quantities can one measure and what are the predictions? And then when all those interesting things happen, those quantities are different.
but yeah, I guess those yeast examples would be good examples of maybe the kind of experiment that you're talking about. Periodically, all the variation is purged. It's like a bottleneck through a single individual if there's no recombination.
The upper cap is almost circumscribed.
Yeah, or background selection or a combination of both. Yeah. Okay, so that's where we are. That's where the numbers are. There's about a 5 order of magnitude range of variation among organisms' effective sizes. There's a gradient, this minus 1/5 power law scaling. It would be nice to know why that is minus 1/5, you know, from first principles. I can't give an answer to that. And I also want to emphasize that there's about an order of magnitude range of variation at any one size of an organism. There's about an order of magnitude range of variation around that. Some's due to measurement error. Some of these populations aren't in equilibrium and so on. But we also expect, you know, parameters like this to range through time for reasons I'll talk about later. So I don't want you to take this too seriously, but this is sort of my cartoon attempt to think about what we call the distribution of fitness effects. Because remember, if fitness effects associated with mutations are sufficiently small, natural selection can't take advantage of them. And so there's been only crude work to try and estimate what this distribution of fitness effects is. This is just be the fraction of new mutations with different deleterious effects in this case. And people have done this in a very, very crude way, usually just thinking about mutations that are really strongly selected for, mutations that are near the balance between selection and drift, and then ones that are just can't be perceived. And I'm just trying to give you an idea, just to get your head, you know, you're thinking about these sort of things. So down here we'd have a small NE species. I guess I'm thinking about an NE of, it's actually fairly big for metazoan of 1 million. And so these should be 10 to the minus 6, if you can see the minus signs there. And so all of these mutations to the right here can be perceived by selection, and all the mutations to the left here in this blue region, those would be in that region of effective neutrality. And then for bacteria, we move this drift barrier way over to the left because we're looking at population size of 10 to the 9th. And there's only this little tiny tail over here of mutations that can't be perceived by selection in bacteria. There are some, maybe a very, very small number of truly, absolutely neutral mutations. About the only ones I could think of that could apply are nucleotide sites in completely non-functional regions, swapping an AT bond for a TA bond. We know how much it costs to make a T and an A, and metabolically they'd be the same. But if you switch a GC to an AT bond, that costs you about, I think it's 5 ATPs in terms of your expenditure. And so knowing the energy budget of E. coli, even those can be perceived by natural selection. I'll probably get to this next class. These are just things to think about. These are things we don't know and we really need to know, is what do these distributions really look like? And the problem here is we don't have any simple way of measuring selection coefficients directly that are this small, because these would change allele frequencies in a lab experiment so minusculely small, it would take, you know, a few generations of scientists to perceive something like that. Okay, now I want to move on to a different way of thinking about effective population size. This is a non-equal approach to thinking about non-equilibrium effective population sizes. And there's been a lot of attempts to do this. People are very interested in asking now whether you can take various measures of patterns of variation in natural populations and infer the past demographic history of the population. Do we have evidence of past bottlenecks or past population expansions and so on? And so I'm just going to talk about one method that I worked out a few years ago with Bernard and Peter. And this uses as fuel for the method. This utilizes information that we call site frequency spectra of populations. And so this is just an example. So what do we mean by the site frequency spectrum of a population? What we mean is that we've gone out and sequenced lots of genomes, accurately estimated the SNP frequencies, the polymorphisms, and here we're focused on silent sites here. And then, sorry about that, let's see. Let me go all the way back to the beginning. And then what we're asking is what fraction of sites have this frequency, what fraction have that frequency, and so on. So that's a site it's the frequency of frequencies in populations. And these data actually come from a study of Daphnia, a Daphnia population. We studied about 100 individuals. And so you get the spectrum of frequencies, and that's it for silent sites and also sites deep in introns. We expect these to be effectively neutral, although I'm going to return to this in a second. And these are more strongly selected sites. So these are amino acid replacement sites. And so you see, they don't get to rise as frequently to really high frequencies because most of them are deleterious and they're being removed from the population. And so we're talking about now what we call the folded site frequency spectrum. We're only going up to a frequency of 0.5. So 0.55 is equivalent, just flipping it over, it's added in the 0.45 bin, because we don't know the derived states of the alleles in this particular case. But there's been lots of attempts to measure these things, and what you need to think about here is that the proportion of sites, polymorphic sites in these different classes, is a function of the past demographic history. If the past demographic history has been one of constant population size, so that our theta for any U remains constant, then this distribution, told to us by Sewell Wright many years ago, will just be inversely proportional to the frequency times 1 minus the frequency. And that dashed line is the neutral expectation for the Daphnia data. So it looks pretty good, reasonably first approximation to the data. And what's important to realize in more complicated data, that every frequency class provides information about different aspects of the history of the population. So on average, singletons, they're only in one copy in your whole sample. These are by definition the youngest polymorphisms on average, and they must give information about the fairly recent past, whereas more highly, assuming neutral alleles, more frequent alleles are giving you additional information deeper into the past. Make sense? To become frequent, you have to have been around for a fair amount of time.
This is from samples of a few hundred.
That's a sample of about 96. We like that number, 96. times 2 because they're diploid.
But if one looks in situations where there's some deep population sequencing, how far down can this?
I'm going to return to this in a bit, and there's some interesting results because I'm going to return in a bit to a sample size of 1,000. And that actually changes things in certain ways for reasons you may know from a colleague. It does, the thing it changes, assuming it's a recombining diploid, the only thing it changes is that the 4 becomes a 2 there. So it's the same basic scaling, though, with the denominator. And this K is just a constant associated with fitting data and bin data. So don't worry too much about that. It's not biology. Okay, so what our challenge here is that we want to use information like this to estimate past population sizes. And in the ideal world, the data, if everything was constant, we would infer a constant NE using these data, but we want to pick things apart in a greater detail. So the approach that I'm going to talk about to estimating past demographic history requires a lot of data. You have to have a large number of genomes because you want to finely dissect that site frequency spectrum. The finer the dissection, the more fine details you can infer about changes in population size over time. And you need to really know your allele frequencies. So you would not use pooled population sequencing to do this because a lot of your rare alleles will just be sequencing errors. So you have to be careful what you're doing. And we use maximum likelihood techniques to estimate that factor out the errors in allele frequency estimation. What we're trying to do is get a model, what I call a model-free method of analysis. So a lot of people try to use information like this and say, okay, let there be a population bottleneck, and let's try and figure out where it was and how deep it was. But we're just going to be model-free here. We're going to just let the demography do whatever it does. And then this is where we get into the coalescent, this thing called the coalescent, and this gets back to the point that the gentleman in the front of the room were making about the relationship between the effective population size and time for alleles to coalesce. And the beauty of the coalescent, this is some theory worked out many years ago by Kingman, and the beauty of the coalescent is you can work out the branching patterns of genealogies and populations without knowing anything about genetics. So the branching patterns are independent of the underlying genetics, underlying demography. That's a sampling process. But the branch links in the genealogy, they do depend on the history of the population sizes. And we're trying to use that information now to infer past population history. So here's the idea, is this is just a cartoon version of a random genealogy. You've got 5 sampled haplotypes in the population. And what you're asking is, let's just take random pairs down here. There's 10 random pairs, 5 times 4 divided by 2. How long does it take to go back if we're just randomly sampling till we get to a node where they're identical by descent in some sense? And the theory's been well worked out here, and that's equal to 2n generations divided by the number of pairs. Okay, so you get your first coalescent there. That's the average time to get the first coalescent in our sample of 5. And now that's coalesced. Now you have a sample of 4, and you plug in the same numbers, and you get that expected time, additional time back. And then you just play the game. You just walk back, and then the sample size gets smaller as you walk back in time. And so you can use this to infer the depth of the different points in the genealogy. Now there's a couple points here, is that usually when people are talking about the coalescent, the population sizes assumed to be constant at all depths of the whole genealogy. And I want to point something out here too that I probably should have pointed out before. The depth of the genealogy is, if you got a large sample size, it's about 4n generations. Let's say 4ne generations. And that means polymorphism data. That means your whole sample will coalesce to one point, at a point on average deeper than that. And that means whatever we're trying to do with polymorphism data to estimate effective population size, there's no hope of trying to infer population sizes 10 NE or 100 NE generations in the past. All the information is based on the current levels of polymorphism. So that was one point I wanted to make, and I've already forgotten what the other one was, but hopefully that'll come up. Oh yeah, this is the other problem. This is just one random genealogy of 5 samples, and the problem is this. Now, if you've only got 2 sequences, there's only one topology. One links to 2, and that's it. If you've got 3, then you've got 3 possible topologies. A, B, C, A's on the outside, B's on the outside, or C's on the outside. But combinatorially, this explodes really rapidly. And so if you've got only 6 sequences, there's almost 1,000 possible topologies, and it goes up exponentially. And here we're talking about almost 200 sequences in our sample. So we got a big numbers problem. So this introduces a lot of combinatorial problems. There's 15, these are the 15 topologies for a 4-sample tree. And so here's what we're trying to do. Again, this is just one realization of a genealogy. And you can imagine this is the form of the genealogy is changing up and down chromosomes, across chromosomes, and so on. So I'm just trying to pictorially show you what's going on as one possible genealogy. And here's all these coalescents. We have 8 samples, and they undergo these various phases. Every coalescent, the number goes down by 1. And so these are like time horizons between the different coalescents. And we're going to let the effective population size vary among these different time horizons. Okay, that's how we're sort of partitioning things up here. And then what we need to know is the probability of mutations arising on these different branches. And so this is the problem. The depth of these different nodes changes randomly over the tree. Singletons always have to be on the exterior branches, right? Because they're the only, a new mutation arises on that branch and only on that branch. And then, as I'll show you in a second, there'll be doubletons and so on here. But we have to calculate this messy thing here that we eventually did, but it wasn't fun, and I'm afraid I'm not going to derive it in front of anybody here. But hopefully you'll get the general principles here. So the point here is that the length distributions of going down here is a linear function of the coalescence going from tip all the way down, no matter where you are on the genealogy. And so we use this information to try and, now we sprinkle down mutations randomly on the tree. Well, not randomly, we know the probabilities of mutations arising on various branches, and we use that information to back calculate what the ends are at the various time horizons, just using the principle I showed you in the simple plot before. And so just as an example, these are singletons. They're all on these exterior branches. Sometimes there's not one on a branch and so on. But we're trying to, we know how frequent singletons are in the population. If you're a doubleton, you would have to be on a branch like this. If you were a mutation there, then 2 individuals in the sample would have that mutation. And then this would be a tripleton down here. 3 individuals in the genealogy would have inherited mutation down here. This would be a quadrupleton, and this would be a quintupleton, if those are words we're allowed to use. So that's the basic idea. We have information on the frequencies of singletons, doubletons, and so on. We have information on the basic topology, the large combinatorial number of topologies, and we're trying to infer the internal branch lengths on those topologies to get a measure of the history of the population. So just to sum this up in words, this is pretty technical, but we're pretty sure we worked out the math, but my head hurt a lot for about a year trying to work out it's a lot of bookkeeping. So just verbally, each frequency class in the population represents a mixture of mutations with potentially unique demographic histories. Some singletons will be on really short external branches, and there'll be rare singleton branches that will go all the way near to the bottom. And we have to take into account all those different probabilities. As I said, the branching configurations are independent of past population history, but the segment lengths are not. The population size at these various horizons tell us how long the segments will be. And these numbers of singletons, whoops, numbers of singletons here I think there's a delay in the pointer for future users. Okay, so what we're trying to do is use the site frequencies distribution to figure out, back calculate the history of population sizes that have occurred under the assumption that the sites we're using are in fact neutral sites. And so I say it's a model flexible fashion because we're not saying that n has to increase exponentially over time or decrease at some rate. We're allowing the maximum likelihood procedures to tell us whether n has actually changed in adjacent situations and so on.
This is all in the small mu any limit.
The small mu any limit.
Theta is always small.
Yeah.
You can't unique. Not all site frequency vectors can be inverted to the time dependent population.
Can you give us an example? I mean, the genealogy applies no matter what's going on mutationally, right? But we are assuming that, well, I think maybe related to your point, if NEU is too high, there's going to be multiple redundant mutations occurring on the tree, and that wouldn't be good. We're assuming each mutation is independent.
Even without that, I thought it wasn't, it couldn't get all site frequency.
Maybe we can talk about it later because I'm not following why that would be.
If your genealogy, that's as your node length for your nitrogen, you can get these modalities in the middle. So, but that's
Yeah, this doesn't rule that out. Yeah. Yeah. The other neat thing about this is if you can figure out the NE at all the different episodes, then you can ask, what's the average age of an allele, average age of all the singletons, doubletons, and so on. You can ask, what's the average effective population size that singletons have experienced over the course of their history, doubletons, and so on. And in fact, get the distribution of this. So when we first worked this out, I think a reviewer wanted us to apply it to real data. So we applied it to a sample from the African Yoruba population where about 100 genomes. Yeah. Well, we are measuring NE at the different episodes. Maybe I should have emphasized it more. We are not going in and saying, let's solve for all 7 of them at once. Because often the whole system will go determined. We're doing maximum likelihood. So we start with just saying, let's assume n is constant. Does that explain the data? And then we shift 1 in and find out if that improves the data fit sufficiently using a likelihood ratio statistic. And then so we gradually build it up.
Relationship, right, between NE and how demographic. So I was trying to see if.
Yeah, well, I think we're getting back to all the stuff I talked about at the beginning of my talk. These are NEs. And I guess if you were brave enough and knew enough about the biology of the organism, you might work backwards and try and turn NE into absolute population size.
So you have NE at NP at 8, 7, and so on.
Mm-hmm.
So if I take a harmonic mean of that.
I see, yeah.
If I say the end of the day.
Yeah.
So that should work, right?
I think so. And that could be used to get at this issue of the average NE experienced by an allele. It's a singleton or a doubleton and so on. I was thinking of an additive average, but you're probably right. It should be a harmonic. I need to think about that a little more.
I presume I can again just say, okay, NE is basically time, and I'm just talking about the probability of coalescence as a function of time depends on the time.
Yeah.
I can think of that and ignore any
Well, you could. I'm not going to ignore it, but because I'm trying to make some statements about demography, but hopefully everybody's getting this issue of time in NE in their heads. Smaller NE means a shorter genealogy, shorter time back to common ancestry. Is that a good way to?
So here's a great real-world example of that. So if you take random individuals from Europe or Asia, the average LD block is about 60 kb, and they have a certain amount of nucleotide polymorphism. If you sample two random individuals from Central Africa, the average LD block is much smaller, and they have a lot more polymorphism. What's a simple way to account for those two observations? Deeper time to common ancestry for two individuals in Africa. Deeper time means more time for mutations to arise. Deeper time means more time for recombination to break those blocks into smaller and smaller individuals. So thinking about time is a very powerful way to think about this. And you can call it time or NE or whatever you want. But it's a nice way to think about a lot of these issues. Otherwise, it can seem a bit confusing.
You just raised a difficult problem. I can't give you a good answer. There's many kinds of polyploidy as part of the problem. I think if everything was independently segregating, if it was a tetraploid, then you just multiply everything by 8 here, but that's generally not the way it works.
Yeah, we can talk about
So,
that. Things like double reduction, all sorts of weird polymorphism.
yeah. Yeah, that's a good point. There's still stuff to be done in population genetics. So, okay, so our first application of the data is illustrated here. I think a reviewer, maybe reviewers in the room, I don't know. But we decided to use human data for the Yoruba population. I think there were about 100 genomes, lots and lots of SNPs. You need a lot of information to do this. Background selection?
Background selection. Because so this corresponds to the variance affected population size. The variance in allele frequency changes with the generations are correlated. So you would expect that to create a signal of a recent increase in population size. And I was wondering how you disentangle that from sort of demographic effects.
Yeah, I hadn't thought about that. I think background selection, as long as it's occurring at a constant rate, I'm not sure that we change anything here, but I could be mistaken.
So if you look at the variance in allele frequency change across different generations, background selection causes them to be correlated. So the variance in allele frequency change over 2 generations isn't just the sum of the variance over the first plus the variance over the second, you have a covariance.
Okay.
And so that means if you look at, sort of if you think of NE1 as being the NE experience 1 generation in the past, or NE2 being coalescence rate between 2 generations in the past, if you have background selection, even at equilibrium, these are not the same. So the long time scales, you kind of have an asymptotic effective population size in this case, but over the short time scales, it creates a signal of changing.
Okay. We'll have to talk about it a bit more, but maybe the good news is these episodes we're talking about are long time scale changes. So anything you're talking about, I think, would be embedded in the longer time scales. Anyhow, let me try to get to this Yoruba study. And I'm not trying to avoid the question, but I need to think about it a little more. So our first application was for this Yoruba population, this African population. These are sort of the data we got. There's ways we can get confidence limits on the demographic changes that I won't go into right now. But the important point is this suggested, I think there were probably, I don't know, 4 or 5 different ends in the whole set of samples. So we're not getting a separate N for every one interval because it's not improving things significantly enough. But one of the things that we just presented this as an example. But, you know, look at this. There's this strong, brief bottleneck in population size. This is a population in Africa down to about 2,000 individuals about 500,000 years ago. We published this paper in G3. Anybody heard of G3? Hopefully somebody. But 3 years later, these guys we weren't really interested in human evolution. We were just using this as an example. 3 years later, these guys published a paper using a fairly similar approach, using a site frequency spectrum to estimate past demography of the human population, and found us essentially the same thing. Results showed that our human ancestors went through a severe population bottleneck of about 1,300 breeding individuals, you know, something short of 1,000,000 years ago. Bottleneck lasted for about 117,000 years and brought us almost to extinction. A sentence like that, I guess, will get you a paper in science. But it was interesting because I wrote these guys and they didn't know anything about our method or our results. And, you know, it was a nice conversation, but it was just kind of an entertaining thing that these guys independently came up with a very similar conclusion. Some of you probably read this paper. I don't know there has to be any selection. It's just a bottleneck. Could have been.
I know, but one doesn't know that it's a bottleneck. When it could be selection makes it look like a bottleneck.
But
Yeah.
You can see a whole chromosome getting gapped, having a signature across the genome. You can get a some major perturbation that messed up a bunch of selection. But I could see a strong chromosome-specific signal for a strong selection.
Maybe that suggests that test, which I guess you looked at, was this consistent across chromosomes?
I can't answer that. I don't remember. But that would be worth looking at for sure, yeah.
But a lot of weak selection is not real fun. That could work out. If you have a lot of weak selection across the chromosomes, we would really get something that's like this.
Well, you've got to have it episodic or something, right? It can't be constant. Why would it give you a sudden trough?
I mean, there's geographical things.
Yeah. Anyhow, that's what the data suggest. I guess we could, if we were thinking, we may have had a chance of having a paper in science.
The important lesson for students, by the way, is there's a lot of fertile ground here for interesting work.
Yeah, you guys are asking a lot of hard questions. I don't have answers for right now. And maybe somebody in the front row does. But I think we're agnostic with respect to what is generating these patterns. These are the patterns.
And to be fair to mind, if you had any sleep in the last 48 hours.
I'm probably better than without sleep than with sleep, but anyhow. We've applied this to our favorite organism, Daphne, as well. And these are just, they have a metapopulation. We've sequenced over 100 genomes from several populations. And this is just a number of them going into the past 3 million years. And I just want to show, I'm just trying to give you sort of a set of examples of the way you can think about these things. And so this is just the average over all of these 8 populations here. This is on the absolute time scale, and what you can't really see there is there's actually a slight trough here. There's a slight decline in the population on average. These are the standard errors across all populations, then the population expands. So, you know, these populations experience reasonably similar demographic histories, and what this is just telling us is that way back until about half a million generations ago. The population was generally on the order, the NE was somewhere in the vicinity of 1/2 to 2 million individuals. That's 500,000. And then there was this slight twofold decline and then a recent expansion. And that recent expansion is at least, you know, these are just sort of stories, you know, you can make up thinking about geological history and so on. But these population sizes expand. All this is happening post-Pleistocene. And this is all happening during a period when North American climate was getting wetter and cooler and Daphne-like water. So it would have been a good situation for them. Okay, so now I'm going to move on to a couple of other points here. And are you familiar with this paper, Daniel? Or maybe not. Maybe you don't remember it. But there's this very interesting.
1 kid and 2 grandkids.
Pardon?
1 kid and 2 grandkids.
Okay, that's right. Who's the grandkid? Ivana? And Ben. Okay. All right. So anyhow, his family wrote a really nice paper a few years ago. I think this is in 2018 or something. They published this paper and pointed out a really interesting aspect of the site frequency spectrum that I think nobody really had anticipated before. The point they made, basically the point to worry about here is there's a lot of information in the lowest frequency class in populations. And by low, I mean below frequency of 0.01. If you don't have a sample size of 100, you're not even going to get estimates below that. And so the point that they made is here, that I'll break it up into phases. So this is a site frequency spectrum like the one I showed before. It's on a little bit of a funky scale here, but it's going way down to frequencies of 10 to the -5. To measure frequencies like that, you need more than 100,000 individuals. So this is way down deep. These are really rare alleles in populations. And this is the expected site frequency spectrum for neutral alleles. And so these guys below this pivot point here are behaving neutrally, really rare alleles. And then there's this break point. It's a function of the effective population size. This U is this is background selection. This is the rate of deleterious mutations in the region of your marker and their selective effects. And so for somewhat more frequent ones, so this is good. These are neutral alleles, but generally we don't know what fraction of our pool in the 0.01 and smaller belongs there. For those above that low frequency, up to this frequency here, the scaling is no longer this inverse frequency scaling. It's proportional to the inverse of the squared frequency. That's because of background selection. And then the third phase here is above this break point. Everybody linked to a deleterious mutation now has been purged from the population. The remaining alleles in this class, now they're again behaving neutrally, but at a reduced effective population size. Well, this is, I think technically this is for complete linkage groups, regions, small regions without recombination. I think that's correct. Okay, so this makes a prediction. This is really nice. This is theory developed. It's like Michaelis-Menten. You know, they developed the Michaelis-Menten equations before we had any real data on enzyme reactions. This is making a prediction about population genetics before we've ever observed this kind of thing. Well, we've now observed this kind of thing in our Daphne work. So I just showed you this before. That's the site frequency spectrum with a sample size of only about 100 individuals. But now this population, we have about 1,000 individuals. And so now we can bring the spectrum down to about, you know, on the order of 0.001. And it exhibits something very close to this scaling. So before I said if you you ignore everything that's going on. This class here is a mixture of everything below frequency 0.01, including the really, really rare ones. But as you increase your sample size, you can start to reveal what's going on within that category. And so what you can see here is these rare ones are scaling. It's not exact, but it's close to 1 over frequency squared scaling, just as predicted by the theory. Pardon?
You have recombination.
Well, the theory is, there is recombination, but the theory is based on regions of linked deleterious mutations where there hasn't been sufficient time to eliminate the associations. Imply what? It depends on the deleterious mutation rate in the region and the selective effects. Yeah. Maybe you can do the math in your head. Those are the breakpoints. So we have rough estimates of big U and S for Daphnia. And this is a point I wanted to make, is that this would be, out here would be that third phase, a return to something like the normal scaling for neutral alleles. This is that second phase. And then the expectation is this would eventually go back to a scaling like this. It's going to take us a lot more sequencing to get down here, but that would be the prediction. Yeah, somebody should do this with yeah, you got to be really careful here, super careful here, because the problem here, all the information that's going to be revealing things is really rare alleles. You got to be sure they're not sequencing errors, yeah. But it would be, I mean, there are few things that have as sample sizes like we have in Daphnia now, but surely it would be nice to get it in some other.
Sequencing errors, if you have a reasonable model of sequencing errors you get from the data, and you can use a lot of the very rare things, even though you're not confident in one. So there's things you want to get.
Well, if you want to factor it out with a model or something on the sequencing error.
I mean, you can develop the models from the data. Sequencing errors don't care if it's not a function.
Well, we do that with our maximum likely estimates. We're factoring out the error rate. And it's not just the sequencing error, it's any kind of error rate. Anyhow,
Since you're seeing this 1 over p squared, the 1 over p squared regime comes in also when there's a lot of positive selection. In that case, it won't go back over the 1 over p. But I think unless you see it coming up like this and then tilting back over, then the pictures with positive selection will be similar.
I hadn't thought about it with positive selection. But if somebody wants to go out and sample this population for a long time, we could resolve this. But the theory at least predicts, right? It's going to be a sort of a sigmoid shaped scaling. And so we'll just have to wait and see. And then this is, as I said, I thought I'd get through that first part of my talk. I'm going to have to scramble and see what other people present over the next few talks. But I think this is where I wanted to end with a quote. But first, this is a caveat, is that it came up earlier in the talk. There are different kinds of effective population size is just a useful way of taking a complex problem and trying to distill it down to one measure that explains a certain kind of observed data. And the NE that I've been focusing on up to now has been what we sort of call the variance effective population size. But an important problem here is that that's the NE that we all rely on generally, because it's the, you can get polymorphism data. It's one way to measure that. And one of the only ways to measure NE, if you don't have, very detailed demographic observations, which nobody does in populations like this. And so I'll get back to this a little bit tomorrow, but it's worth at least thinking about, those of you interested in theory, is, you know, what is the right NE to use in different contexts? Like there's a fixation thing we call the fixation NE that dictates how probable it is that a mutation will fix. That's not necessarily the same NE as this here. It's probably not going to be off by an order of magnitude, but still we need to be a bit careful here. Okay, so I'm just trying to get you the general concept of all these things across today. There's lots of room for work here. And I thought I would end with this. Everybody loves Darwin quotes, right? And I think I have found that theoretical population geneticists are really fond of this quote here by Darwin, that he just knew we were the ones, or you guys are the ones, you guys that are better theoreticians than me. Mathematics seems to endow one with something like a new sense. If only I could do math. But then he wrote this note to one of his friends who was a mathematician. I suppose you are 2 fathoms deep in mathematics, and if you are, then God help you. For so am I, only with this difference. I'm stuck in the mud at the bottom in the primordial ooze, and there I will remain. If there's any ambiguity at this point, here's another quote from Darwin. A mathematician is a blind man in a dark room looking for a black cat which isn't there. Thank you. I guess there's a few minutes for questions. I think questions are going to be helpful for us early speakers to know what's registering and what's not. And
Actually, maybe a show of hands. So how many you are, you can vote multiple times. How many of you would consider yourself a biologist? How many would consider yourself a mathematician? A geneticist?
they read the last quote. They were.
There's a black cat here somewhere. An evolutionary biologist?
Physicist.
Physicist. Here we are.
Okay. What's the conclusion?
Well, just think of this. If they're all physicists, we speak differently if they're all biologists. So. But we want to encourage you to keep, and have been bashful, but by the end, have been bashful about asking questions. That's important because that helps us let us know where you stand relative to what we're trying to convey.
Of course, we're not monitoring the people on, there's a bunch of people on Zoom, aren't there? Or is there just 2 or 3? Or are there like hundreds or thousands on Zoom? Fred's on there. I don't want to touch it, so I don't know how many are on there. Okay, well, thanks a lot. I'll give you an extra couple minutes for coffee. There will be coffee, won't there?